Hi everybody, this is Miss Peachy with your WCA biology class, and this is actually take 2 of a video that I'm doing on viruses and bacteria. So I just want to kind of go through the basics with viruses and bacteria, and hopefully this will help bridge that gap since we do not have any virtual classes this week due to ACT testing. So first of all, we're going to start with viruses. And viruses they're not technically considered living things because they are not cellular. Remember, one of the main kind of characteristics of living things is that they are made up of cells. But viruses don't have cells. They do have genetic material. They have a nucleic acid, either ribose nucleic acid or deoxyribose nucleic acid, RNA or DNA inside of them, and a protein coat. So that's basically the structure of a virus. Viruses can be many different shapes, many different sizes. They are much smaller than the smallest bacteria, but they come in all sorts of shapes and sizes. This is a bacteriophage. This is a virus that actually infects bacteria. So viruses can infect not just humans and mammals and animals. They can infect plants, they can infect fungi, protists, even bacteria. As you can see here with this bacteriophage, it is made up of a protein coat. Kind of looks a bit like a lunar lander, has a bit of a weird structure to it. Inside of that bacteriophage, there is a strand of DNA found inside the head of the bacteriophage. And when that bacteriophage actually lands on an E. coli bacteria, it will inject that strand of DNA into the bacteria. And that's how it gets its genetic material inside of it and how it reproduces. There are 2 main ways in which viruses reproduce, either using the lytic or the lysogenic cycle. And different viruses, there are different types of viruses that can do either one of these, like one or the other, or some of them actually do both. The lytic cycle, the first one, the lytic cycle, is a very kind of a shorter, more, you get sick quite quickly after the infection occurs. So once you get infected with a bacteria, once you get infected with the virus then hijacks the cellular machinery, takes its strand of DNA, and starts to use the cell, all the enzymes and stuff in the cell, to transcribe and translate the information stored on the strand of DNA from the virus itself. That strand of DNA tells the cell to make more viruses. So it does, makes more viruses. Eventually makes so many viruses, the whole cell just splits apart, releasing all the new viruses into your bloodstream. Those new viruses then go on to infect new cells. And each virus infects another cell, making a whole host of brand new viruses. Lucky for you and me, we all have immune systems. And we'll talk more about immune systems coming up here when we look at animal biology. But that helps to mitigate this attack so that not every virus we have will cause our death. The lysogenic cycle is much more insidious. It reminds me a little bit of the Trojan horse. It sneaks inside of the cell, and you don't even know you have it until you are already very, very sick. Lysogenic cycle, again, the virus infects the cell, injecting its DNA into the cell, but instead of hijacking the equipment of the cell to make more copies of it right away, it actually just incorporates itself into the host cell's DNA. So it becomes part of your DNA. As you make more copies of your cells as you age and time goes on, that host cell, every new host cell has part of that old viral DNA still incorporated in it, and it spreads throughout your body. some environmental factor eventually will trigger an infection. So that lysogenic cycle then turns into a lytic cycle. And at that point, then the DNA that's in your own DNA, the viral DNA that's incorporated into your own DNA, starts to become decoded. And new viruses are formed at that point, and the lytic cycle continues where copies of those viruses are made, and then those viruses are released from the cell. But instead of having just one cell that this happens to and having it be kind of a spiraling effect, it actually happens all over your body at once. So a couple of things here to think about. First of all, in the first example, where viruses reproduce only with a lytic cycle, you tend to get sick very quickly after the infection occurs. You tend to start off being kind of sickish and getting more sick, right? And then your immune system attacks it and then you get better. So that pretty much kind of parallels what you would expect with the flu or a cold of some kind, that kind of thing. With the lysogenic cycle, because you already have a full-blown viral infection before you even show any symptoms, it actually is oftentimes a lot harder for your immune system to get to right away, and you can get pretty sick from that. Also, because your body is making copies of this viral DNA over and over and over again, a copy of a copy of a copy, there is more room for genetic mutations. So oftentimes you end up with, a very slightly different version of what you were originally infected with. An example of a virus that uses the lytic or the lysogenic cycle would be the herpes zoster virus. If any of you know anybody or have yourself been infected with shingles. Shingles actually occurs because you were one time infected with the varicella zoster virus, which is for chicken pox, right? And that went through the lytic cycle, but then the DNA was incorporated into your cell DNA and made copies of itself, and for years just lay dormant in your body. And then some environmental trigger, stress, illness, aging, triggers that, causing a full-blown shingles infection. The other type of virus we didn't talk about is something called a retrovirus. And a retrovirus is slightly different from a regular virus because instead of having DNA as its genetic material, it has RNA. And RNA can't directly infect a cell. So the first thing it needs to do is it needs to change back into DNA. So retroviruses contain an enzyme called reverse transcriptase. And the reverse transcriptase actually takes that RNA, changes it to DNA, and then it infects the cell. And then when it infects the cell, that DNA can either go through the lytic or the lysogenic cycle, depending on how that particular retrovirus reproduces. Some retroviruses, well, let me just go back to say that this process of changing RNA to DNA means that there's an extra copying process happening. And every time you have an extra copying process, you have some room for error and for mutations. So retroviruses tend to mutate a lot more than other viruses do, which is why they're really hard to treat. Examples of retroviruses would be HIV, and HIV, is a retrovirus that uses the lysogenic cycle, just lays dormant for quite some time before you become infected with AIDS. But another example of a retrovirus would just be influenza. And as you and I both know, influenza changes every year, right? That's why when they have a vaccine for the flu, it isn't always effective. Because it is a retrovirus, it's constantly mutating. And it's mutating in your body multiple times, and it's mutating in everybody's body multiple times. So that means that even if you have the flu and your mom has the flu or your brother has the flu, you might have slightly different viruses because it has mutated even, at that rate. So you might have a slightly different mutated virus compared to somebody who's even living with you, because retroviruses tend to mutate very, very quickly. So that's kind of like the lowdown on viruses. They're not living, but that's debatable, right? I mean, according to our definition of what it means to be alive, they are not alive. They are made up of some sort of nucleic acid, DNA or RNA, and a protein coat. They infect the host. They need the host for reproduction. They kill you, basically, or hurt you or make you sick because they want to reproduce. That's what they're using your body for. They reproduce either using the lytic or the lysogenic cycle. And viruses that use the lysogenic cycle have more chances of mutations. So they tend to be a little harder to treat. But they also, because you have a full-blown infection before you're even showing symptoms, they also tend to make you sicker. Also, retroviruses, which have RNA instead of DNA in them and have to change first to DNA, mutate more quickly and therefore are much more difficult to treat as well. The other thing in this unit we're talking about is bacteria. Now, bacteria can be divided into two groups. You have eubacteria and archaeobacteria. And in fact, and just pardon me for a second, because speaking of viruses, I have a little bit of a cold here, but and in fact, just a few years ago, they changed like how the kingdoms of living things are organized to include these archaeobacteria, because they're so different from regular bacteria that they've become their own domain. They're just their own group of living things. So we have the eukaryote, and then we have the eubacteria and archaeobacteria, which are different from each other. Both are single-celled organisms. Both are prokaryotic cells. Remember that prokaryotes means the cell's really small. It has a capsid. It's like a cell wall, right? It's a capsule, and it has a cell membrane. It has cytoplasm. It has DNA inside of it and ribosomes, so it can still do protein synthesis, but it's just really tiny. Compared to a regular cell, like the cells in our body, it's about the size of a mitochondria. It's really, really small. So both are prokaryotic. They don't live in groups or anything like that. The big difference between the two of them is that archaea don't have this protein in their cell wall called peptidoglycan. So peptidoglycan is absent in archaea, but it is found in bacteria. Another thing that makes them different, bacteria or eubacteria are sensitive to antibiotics like penicillin or amoxicillin or erythromycin. Archaeobacteria are not. Bacteria are pathogenic. They infect human beings, make us sick. Archaeobacteria do not. And archaeobacteria also tend to be called extremophiles. They live in extreme environments, like very, very hot places, such as like the hydrothermal vents in the bottom of the ocean or inside of a volcano, or very salty places like Great Salt Lake. I mean, places we thought that living things could never live before they have been found. Archaeobacteria are actually thought to be the most ancient of all prokaryotes because the Earth was at one time a very hostile place to live, and archaeobacteria would have been able to survive in such conditions. Regular bacteria, eubacteria, excuse me, they have harmful relationships. We call that saying that they can be pathogens, but they can also be very helpful. I mean, you have all sorts of bacteria found in your gut. You need those to be able to survive. You would be very, very sick if you didn't have those bacteria found within your large intestines. So we're going to look mostly at eubacteria. Eubacteria, there's like 3 main shapes that make up eubacteria. Spherical, which are called the cocci, rod-shaped bacillus, and spiral-shaped spirillum. And you can see actual electron microscope pictures of them here. You can see some other pictures. This is Bacillus tetani. This is a rod-shaped bacteria that causes tetanus. Streptococcus pyogenes, which I spelled streptococcus. I spelled that incorrectly here, but you can see the spherical shape of the cocci there. So that, I mean, you'll hear these terms if you get sick or something. You might hear that you have strep, and that's where that comes from. Also, bacteria can be categorized as being either gram-positive or gram-negative. I guess both gram-positives and gram-negatives can cause human disease, but gram-negative tends to be more bad, harder to treat. The big difference between the big difference between them, or how you identify them, is you do something called a gram stain, which is a type of technique where you take all these different dyes and stains and you put it on your sample of bacteria. And if it's a gram-positive, the bacteria turns purple, and if it's gram-negative, it doesn't. And that's the distinguishes between a gram-positive and a gram-negative. Gram-negatives, like I said, tend to cause more human disease and human diseases that are more harmful, due in part to the fact that their cell walls are harder to penetrate, so it makes them harder to kill. Okay. Cells, or these bacteria, can move. They can move using flagella, which helps them, kind of propels them around in a whip-like motion. They can use cilia that helps them to kind of swim around, or they aren't moving at all. They can be non-modal. Some examples of what flagella would look like, or cilia would look like. Some bacteria are autotrophs. They make their own food. Or chemotrophs. These also make their own food, although these are much lesser known. They're often found in the depths of the ocean near hydrothermal vents, and some of them can be archaeobacteria as well. But autotrophs would include things like cyanobacteria. These are also called blue-green algae, but they're actually a bacteria that lives together kind of in a colony in a way, and they're believed to be one of the oldest of all bacteria because they were responsible for producing a lot of the oxygen in the ancient Earth's atmosphere. Some bacteria are heterotrophs. They actually eat other things, and many bacteria are considered to be decomposers. They will eat dead things and break them down into their fundamental nutrients to recycle into the earth. Bacteria will reproduce using either asexual, or it's primarily asexual reproduction, but conjugation is kind of a form of kind of like sexual reproduction for bacteria. Fission, they're just splitting apart. It's you copy the DNA and it just splits apart and makes a complete clone of itself, right? Conjugation is where two bacteria actually come together and they swap some genetic information. So it's kind of like sexual reproduction for bacteria, and it's how bacteria maintain genetic diversity. It's how, information is shared. It's interesting, it says here, such as resistance to penicillin. So it's one way in which you might have, like if you have a bacteria that has some sort of antibiotic resistance, that can be passed along to other bacteria that don't contain that gene for that resistance to antibiotics. And we also have spore formation. This doesn't happen with all bacteria, but some bacteria can form dormant spores that just allow them to survive inhospitable conditions, like if the environment gets really, really harsh, so that they can still live and come back when the environment becomes more favorable. Bacteria can live in symbiotic relationships with organisms. We have symbiotic bacteria in our large intestines right now. They help us because they break down food that we can't eat, but they also produce vitamins that we need, like vitamin K is produced in our large intestines. So those are very, very beneficial and important to our health. That's one example of a symbiotic relationship. But bacteria can also be parasites. They can actually cause disease and live inside of a host cell, injuring them. An example here would be with the bacteria that causes tuberculosis. Another example of a mutualistic relationship, a beneficial relationship, is that with plants that is known as nitrogen fixation. So plants actually have certain bacteria that live sometimes in their roots, sometimes it's a fungi that does it. There's various types of organisms that work with nitrogen fixation, but they take nitrogen from the atmosphere and change it into nitrogen that plants can use and is used by all living things. And finally, the last thing we talk about here is something called the endosymbiotic theory. And the endosymbiotic theory is basically, in a nutshell, it's explaining how eukaryotic cells came into being today. Is that there used to be two different kinds of prokaryotic cells. One that was a heterotroph, right, that didn't make its own food, but it also needed oxygen to survive. So it was a aerobic bacteria. And one that was an autotroph that was able to do photosynthesis. And if one bacteria actually engulfed the other one and took that bacteria inside of it, then they lived in this mutualistic relationship. The bacteria that did photosynthesis could provide food. The bacteria that was an aerobic bacteria could use oxygen to break down the food, and they would have this kind of relationship where both would benefit. And over time, since bacteria can exchange DNA using conjugation, they were able to trade DNA with each other so that new bacteria would actually contain both types of bacteria, and that was like the beginning of the first organelles that were found inside of cells. So instead of a cell just being a empty, just had some jelly inside with some DNA, it actually has these internal organelles, which was the first eukaryotic cells. All right, so eukaryotes and prokaryotes are have very many differences, but they also have some similarities. For example, both have ribosomes, both have DNA, both have cytoplasms and a cell membrane, and some have flagella. And of course, the differences are many here, especially and probably most importantly that prokaryotes don't have any organelles at all. They are small and simple. All are unicellular, but they are believed to be the first cells that were found on Earth. All right, so that is the end of today's little summary on viruses and bacteria. If you guys have any questions or concerns, you know where to find me. You can give me a call or send me a webmail message. Have a great day, everybody.